Hello, everyone, and welcome to this session. It's my tremendous pleasure to introduce a good friend of mine, Marc Howard, who's a professor in the psychology department at Boston University. And he's going to tell us about some of the work he's been doing lately to understand how the brain uses time cells to think, represent space, time, and memory. So without further ado, I'm going to turn it over to our distinguished keynote speaker. Go ahead.
Thanks, Brad, and thanks to everyone who's worked on organizing this online conference. It's really exciting to see people sort of reinventing ways of how we do business as academic scientists, and it's really great. So go team. So I have a confession to make, and that is that I'm not really a neuroscientist. I started in cognitive psychology, and I've worked in mathematical psychology and cognitive science. And I've sort of come into neuroscience sort of necessity. The reason I've come into neuroscience is an attempt to place physical constraints on theories of how we might think and remember and do things. And in this talk, everything I'm going to show you is going to be motivated by this theoretical perspective, but I'm not really going to be able to go into that in any detail in this talk. So this might seem a little nuts. But I encourage you to look into other work that we've done as that's provided inspiration and background for this. And I'll allude to some of this as we go along. So about 10 years ago, inspired by ideas from really the dawn of psychology, William James, for instance, talked about this a great deal. My student Kartik Shankar and I worked out a hypothesis for how the brain might maintain information about the recent past and how that information might be used to construct cognitive models of a variety of memory tasks. And here's the basic zero equation hypothesis. So as an event happens, as something unfolds over time, we listen to, for instance, a melody, boop, 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 something like that. As it recedes into the past, neurons in our brain actively maintain a record of that recent experience. And they, much like the musical staff going diagonally in this figure. And so that at each moment, we have a record of what happened when in the recent past, and that different neurons ought to be responsible for different parts of the past, so that our memory is organized like a timeline of recent experience. And so moreover, in addition, very much inspired by, again, very long-standing ideas in cognitive psychology, we proposed that the resolution of this temporal axis ought to be compressed, such that you have a less good idea of when things happened further in the past than more recently. That we have a good memory for the recent past and more fuzzier information about when things happened in the past. And so you can operationalize this as the brain contains a population of neurons with receptive fields organized along a time axis, right? And one population of neurons is responsible for things that happened maybe 1 second ago, and another population is responsible for things that happened 2 seconds ago, or 4 seconds ago, or 8 seconds ago. Okay, so that's sort of where we started. Around the time we were making this proposal, people discovered a phenomenon called time cells, and people here mostly Yuri Bushaki's lab, Eva Pashtelkova in Yuri Bushaki's lab, and Chris McDonald in Howard Eichenbaum's lab, and the great Howard Eichenbaum did a number of really important studies on this. But they observed a population of cells that behave kind of like what we were looking for. And so let's think for a moment and think about how a population with receptive fields in time would behave as something as an event recedes in the past. So if some triggering stimulus comes, clap, as it recedes into the past, it should enter and then leave the receptive fields of different neurons, different time cells. And so if we're observing from the outside, what we should see is neurons that fire in sequence, that fire at a consistent sequence across different occurrences of the same stimulus. And on the left here, I'm showing you three very nicely chosen hippocampal time cells from dorsal CA1. And here I can show you a population, also from the Eichenbaum lab, a population of 172 simultaneously recorded neurons sorted on their peak time. Okay, and so this population, I hope you can convince yourself of quite quickly, carries information about when the stimulus that started this sequence began. Right? By noting which cell fires, I can figure out how far in the past that thing was. I'm going to tell you about a couple of more results from work that various work showing that different stimuli can trigger different sequences, such that if we have a boop and a beep, we'd get different sequences of time cells. And so by noticing which cells are firing at any particular moment, we can reconstruct not only when the stimulus happened, but also its identity. I'm going to argue that the time axis, as we proposed a while ago, is compressed such that it doesn't have equal accuracy for different parts of the temporal past. I'm going to argue it's probably a logarithmic compression, although you could fight about that. You can actually appreciate really rapidly that the time axis ought to be compressed to the extent that you appreciate that there's a curvature here in this population of cells. If the receptive fields were evenly spaced on the time axis and we sorted the sequence on its peak time, we'd see a straight line. The fact that we see a curved line like we do in this figure means that there's many more cells with receptive fields at the recent past and fewer as they go along. And we'll formalize that in a minute. And I'll also emphasize, although this phenomenon has received a lot of attention in the hippocampus, which is presumably responsible for episodic memory, we see kind of the same phenomena. We see very similar phenomenon in a number of other brain regions that are prefrontal cortex and striatum that are presumably responsible for working memory and implicit memory or conditioning, however you want to define that. Okay. Okay, so I'm going to go through a series of studies here and sort of convince you of these points in more detail. This is a paper from recordings. Recordings were done by Earl Miller's lab, originally published in a 2010 Neuron paper. We're looking here at reanalyses by Zoran Tiganj, who's currently in the Department of Computer Science at Indiana University. And in the task, the monkey has to remember what stimulus began the delay. So it's a working memory task, and the animal gets either a dog or a cat, a picture of either a dog or a cat, and then they have to wait, and they get another picture, and they have to say whether it was the same category, yes or no. And there's two categories. There's two types of categories. There's cats and dogs, and then there's cars and trucks. And to the extent we can figure out, to the extent we can decode which stimulus was at the beginning of the trial from looking at the firing rate during the delay, we can conclude that this population has information about what happened when, much like the different notes on the staff and the figure I showed earlier. So the first thing I want to establish is that there's time cells in this data set. I'm showing you a plot of the active cells during the delay interval as a function of time, and hopefully this looks to you similar to the previous such plots I showed with time cells before. But moreover, the activity depends on not only when the stimulus happened, but also the identity. And so in order to evaluate this, what we did was we took each neuron and we sorted it on the basis of its best stimulus. So let's say the best stimulus is a dog. And then we compared the firing for the same cells with the same sort to the other stimulus from the same category. So if that was dogs, dogs are the best. And then this is cats, also from the same type of domain. We see kind of a similar temporal curvature. However, the same cells fire much, much less to cars and trucks. So we can see, meaning that cells respond best to one particular type of stimulus, and their firing gives us information about identity within the category as well as the different categories, such that we can look at firing during the delay interval and construct a decoder and decode well above chance whether this trial began with a cat or a dog or a car or a truck. And in this analysis, and many people have done this, so hopefully this is familiar, we trained a classifier on a segment of time, say a 50 millisecond bin, say 0.75 seconds after the stimulus, and then we tested that decoder on each of the other time points. And we find that the decoder is above chance, indicating that that the population has information about what happened, but also it decorrelates over time. And that's a consequence of the fact that there's sequences of cells, that there's also information about time in this population. And so I chose this study because we had it handy, and it's especially nice. But we've also seen the same results from monkey PFC and hippocampus. Nathan Cruzado has a paper, should be out in Hippocampus briefly, that uses the same type of analysis on a different kind of task. And other groups have seen this as well. Yanis Tashidis from Payman Golshani's lab just recently published a neuron paper showing the same type of thing from rodent hippocampus during perception of odors. Okay, so it seems that this population of time cells carries information not only about when the triggering stimulus took place, but also when it took place, as we propose. Okay, so I want to talk for a minute about the compression hypothesis. Actually, let me go over to here. So we here, meaning neuroscientists, have a lot of evidence that the brain represents continua out in the world along a logarithmic scale, okay? What I'm showing you here is a very famous set of figures from the visual system, where we're plotting size of visual receptive fields on the y-axis as a function of distance from the fovea on the x-axis. And you see that rather than cells throughout the visual system rather than receptive fields throughout the visual system having the same characteristic size, they get wider as you go to the periphery. And this has been understood for a very long time. And you see a cartoon down below. It's also been known for quite some time, dating back to the 80s, it seems that we resolved this pretty well, that the compression of the visual system, at least outside of the fovea, seems to be as if the receptive fields are evenly spaced on a logarithmic axis. So let me go back to this other picture. So if we had receptive fields that were evenly spaced and evenly tiled a logarithmic axis, we would see, when we looked at them as a function of not log, there's the regular retinal position, we would see that as we go away, the receptive fields get wider and less numerous, right? And we believe that this phenomenon, this logarithmic compression, supports, at least in some circumstances, the Weber-Fechner law, which is a very, very old result from psychology, and holds for nonverbal numerosity as well as vision. And we proposed that this ought to be this same sort of compression ought to hold for time as well. Okay, so we're going to try and evaluate that. And so here I'm showing you some preliminary results from recordings by Jay Bladen in the Eichenbaum lab, with analyses being done by Ray Tao in my lab. And what Ray did was she estimated, first of all, you observed time cells in this experiment, as one would expect. What she did was very smart. She estimated the receptive fields rather than averaging across trials. She averaged the receptive field on each trial and estimated the distribution of locations using hierarchical Bayesian analysis. Okay, and so you can see 3 example cells, much like the ones I showed it earlier from Chris McDonald, with this fit taking place, and you can see the little squiggly lines. The width of the receptive field is now estimated within trial, and we can separate that out from the across trial variability. And if there's logarithmic compression of this time axis, we would expect a linear relationship between that within trial receptive field width and the location across trials. And C here shows the width of the within trial receptive field as a function of its location. And to the extent you accept that's a straight line, you accept that there's logarithmic compression. She also estimated the number density of cells. So I've been saying for a while that this curvature is consistent with the compression. It turns out that a logarithmic compression has a very specific form, and we should be able to express, if there is literally a logarithmic compression of the timeline, we should see that the number density is coming from a power law distribution with an exponent of 1.0. And she estimated, she fit the distribution of means across cells, again, using hierarchical analysis, using a hierarchically constrained analysis. And the inset here on the left shows, and hopefully you can see it, shows the posterior distribution of that alpha, and it overlaps, it lands exactly on 1.0. The little vertical line hits 1.0, so that's a good job. So it seems that we certainly can't falsify the hypothesis that the compression is logarithmic, and there's strong theoretical reasons to think that ought to hold as well. So let me sum up what I told you about time cells just now, and then I'll tell you about some other stuff, which is also really cool. So time cells fire in sequence, and they carry information about the time since the event that triggered the sequence, okay? We know that different stimuli trigger different sequences, carrying information about the stimulus, the event that triggered the sequence. Because of these two properties, we can decode what happened when from this population. We're certain that this that the temporal axis is compressed. It's not, there's decreasing accuracy for events further in the past, and we think it's probably logarithmic. And I would also add that I showed you results from hippocampus and prefrontal cortex. Other people have observed similar phenomena from the striatum, and that was also part of our original proposal, that this memory for the past, this sort of experience of the recent past with what happened when, ought to be important in many different forms of memory, including working memory and episodic memory and conditioning as well. And there's pretty good arguments for that that I can't go into today. Okie doke. All right. So that was part of the stuff we wrote on the chalkboard 10 years ago. We spent a while trying to figure out how you could generate a population of cells that coded for the past with this logarithmic compression. And we could only think of one thing. And once we thought of that, it seemed like a really good idea. And so we've pursued this for a while. And I'm going to tell you basically about recent evidence for this other kind of coding. So we proposed in the 2010 paper and subsequent stuff as well, that there's not just time cells. The time cells are derived from this other population of cells. Let's call them temporal context cells. And rather than having receptive fields that tile the time axis with different cells having different peaks along different parts of the axis, we proposed that these cells ought to code for the same type of information with exponential receptive fields, and that rather than having different time points, rather than peaking at different points like those Gaussians I moved along, that the variability across cells should be carried by the rate at which they decay. Okay, and so here are temporal receptive fields for, as a stimulus is a certain amount of time in the past. One cell might have a fast time constant, and other cells might have slower and slower time constants, but there's this family of receptive fields that have exponential decay that would also be used to generate time cells, okay, that carry information about the past. This is also the real Laplace transform of the past. If you had a population of cells with spectrum of rate constants, the reason we know they would be coding information about the past is they're the real Laplace transform. But we can omit that for a moment. Let me try and take a moment and sort of convince you that a population of exponentially decaying cells with different rate constants S you'd be able to figure out when a triggering stimulus took place using that population. So first, I'm showing us our old friend, an exponential curve. I've chosen the rate constant, s, and the time constant, 1 over s, or tau, to be equal to 1. And this is an exponential curve that hopefully everyone's seen a bazillion times. Okay. If we zoomed in on this function, so if we looked at times much, much shorter than 1, we would see that this cell is not conveying useful information about that time. I've just taken exactly the graph I just showed you and zoomed way in on the x-axis. Similarly, if instead of zooming in, if I was looking at times much, much larger than 1 second, like so, and I've just taken exactly the same curve and zoomed way out, the cell is also not conveying useful information about the time at which that event took place. The firing for time 100 and time 150 and time 200 and time 250 is not different for the cell, it's identical. What the cell is doing for you, though, is telling you good information around the time scale of its time constant. And basically what it's doing is giving you the ability to tell whether the triggering event was earlier or later than its time constant. So if I had a lot of cells, here I've drawn, hopefully you can see this, 3 cells with 3 different time constants, red cell, blue cell, and green cell. If I look at this population and I'm able to simultaneously observe all of the cells, and I find that, for instance, red cell has turned off, but blue cell has not, I know that the time of the event must have been somewhere between the 2 time constants of those cells. Similarly, if I observe that green cell is still firing, but red cell and blue cell have turned off, I can figure out that the event was somewhere between the time constant of blue cell and green cell. And by having many cells turning off in sequence, I can figure out how far in the past the stimulus took place. There's like an edge as a function of time constant of these cells that I can use to infer how far in the past the triggering event took place. And that's the basic idea. More formally, we can, and this is sort of where we started with, more formally, we can say that a population of cells with exponential receptive fields, that's all I'm showing you right here, f of s, is just a population of cells with a set of exponential receptive fields with different values of s, being convolved with being activated by the actual history, taking into account the actual history. That's the real Laplace transform of f of t. So the real past is related to the Laplace transform in a one-to-one relationship. And we can know, much like a distorted mirror carries information about the image, but need not look exactly like the image. That's my excuse for showing you a picture of Mo Howard. In addition, we know how to invert the Laplace transform, so we can imagine a different set of cells constructed by an attempt to invert the Laplace transform, and then it turns out they would look like time cells, right? So the idea is that time cells behave like an attempt to invert the Laplace transform, and that there's this other population that codes for the transform. And so to summarize, for the last decade or so, we've proposed the following theoretical framework where some event happens, click, some f of t happens at some point in time, a bunch of cells activate at about the same time and then turn off at different rates. And that's used to generate a set of basically time cells that have receptive fields in time that are compressed. And I'm just in the middle column here, I'm showing you the heat maps you'd expect from a population of such cells. And I've chosen, and we've thus far seen a ton of evidence in this talk and also elsewhere for populations of cells that look like these attempts to invert the Laplace transform. But until quite recently, there wasn't evidence for this other population holding the Laplace transform. Okie doke. So, but that has changed. Over the last 2 years, we've seen quite strong evidence for this. I'm going to focus on a paper that's out in PNAS over the summer from students Ian Bright, Miriam Meister, and Nathan Cruzado, reanalyzing data collected by Miriam in Beth Buffalo's lab. And the monkey we're recording from the entorhinal cortex, and the monkey just sees a picture every so often. And we're syncing to the time at which the picture is presented, and that's it. And so a bunch of pictures are presented. And I'm showing you 3 cells that, rather than firing sequentially, all change their firing. Most of them are excited, but there's some that are inhibited at around the time the image is presented. And then they relax back to baseline with at different rates, with different time constants. Okay. And so here is a plot of all of those cells sorted like one would, sorted in much the same way one would for time cells. And rather than seeing this J that tiles the interval, we see essentially a vertical strip, because all of the cells across the population activated about the same time, and then they deactivate at different rates. And for funsies, I'll just remind us what the model picture would look like, and then quickly go ahead and you can convince yourself that does or doesn't look the same. Okay. We can also decode time, and we did a bunch of methods and stuff, and you're welcome to, happy to look at the paper, and I'm happy to answer questions about in the Q&A. And also, we know that you can decode time from this population. If we ask, if we train a decoder to try and guess how far in the past the stimulus took place, we can figure that out. And this is a temporal decoder. To the extent there's a bright line down the center, you're convinced that the decoder is able to decode time. All right. So let me take a moment. So thus far, and there's a paper by Albert Sau in the Moser lab showing essentially the same results for rodent LEC from 2018 in Nature. And so we're pretty excited that that was confirmed. And so far, we've basically caught up to the theoretical proposal from a decade ago. In the last decade, since we wrote that stuff down, we've continued thinking about cognitive science and cognitive psychology and computational stuff and why this might be useful. And I at least have become convinced that this is a really useful idea for like a theory of brain cognition. And I don't have time to convince you of that in this talk, but I'm taking this very seriously. And I don't think you're doing a bad thing if you also take that seriously. To give you an opportunity to try and take that seriously as a model of many things, I'm going to show you really super quick a study on a completely different domain. So this is Rifki Afan reanalyzing data that's previously published from Carol Svoboda's lab, 2018 Journal of Neuroscience. Inagaki, he's the first author. And this is a motor preparatory task. And what happens in the experiment is the rodent gets a tone, boop or beep, and then they have to make a response later. They have to withhold a response for some period of time. It's an easy decision. If you get the low tone, you're rewarded for licking to the right. And if you get the high tone, you're rewarded for licking to the left. But you can't respond yet. You have to wait. So rather than like a beep receding into the past, there's this planned action of a future lick that's at some point in the future that comes towards you over time, right? You have to plan on being able to make this lick at a particular moment in the future, and you have to wait. And what these authors and other people have observed is that during the interval where the animal's waiting, these cells in ALM, anterior lateral motor cortex, ramp up over time as if they're growing towards the bound. As if they're ramping to the time at which the motor plan is going to be executed. And you can decode well above chance which, I think this is what I'm showing you. You can decode well above chance which movement the animal is going to make. Whoops, oh no. So what Rifki did was he took the analytic tools that we developed for entorhinal cortex and just applied it to this data set, which is very generously shared on crcns.org. And if you have a set of cells coding for the time since something happened, I'm just recapitulating results from the entorhinal cortex on the left here, you should see this pattern like so. In contrast, if you have a population that's coding for the Laplace transform of the time until something is going to happen, right? It should look like a mirror image of the results that we saw from entorhinal cortex. And I've shown you in the examples, I showed you ramping cells that had a variety of time constants. Here I'm showing you sort of basically the same idea. To the extent you accept that there's a mirror image between the left side of this plot and the right side of this plot associated with the change in sign of the remembered time, here it's positive, here it's negative, you accept that this population holds the Laplace transform of the future. So that's work in progress, but I think it's really unlikely that this would have worked as well as it did unless there was something to this idea that the Laplace transform is good for many things. All right, so to summarize, we have, I've shown you results where time cells in the hippocampus estimate functions over the recent past. They're compressed, etc. I've also shown you results that these temporal context cells in entorhinal cortex behave as if they're estimating the Laplace transform, real Laplace transform of the objective past as functions of time. I also showed you some preliminary results suggesting that this Laplace inverse pairing, well, first of all, the fact that the hippocampus looks like the inverse and the entorhinal cortex looks like the transform, that places really strong constraints on where time cells come from. Time cells receive input from a Laplace transform of the recent past and behave as if they're an attempt to invert the Laplace transform. I've also showed you evidence that other brain regions, in this case ALM, at least use this idea of Laplace, that behave as if they're using this idea of Laplace transform to code for other variables. Okay. And so to summarize this whole thing, I think this hangs together really well. I haven't really talked much about the psychology and I haven't really talked much about the theory, but there's really good reasons to think that this Laplace transform is extraordinarily useful computationally. There's a paper by me and Mike Halsamo that's on arXiv right now that I encourage people to take a look at if they're interested in getting a high-level overview of these ideas. This is really general and I'm quite excited. Here's people whose work I highlighted today. Zoran Tiganj is currently a faculty member at Indiana University. Ray is a postdoc in my lab. Jay Bladen is currently used to work with Howard Eichenbaum, is currently a postdoc at Brandeis with Shantanu Jadhav. Steve Czerzinski is applying to graduate school. And so if you're looking for a PhD student, Stephen might be the one you want. Ian Bright, Miriam Meister took the co-lead on that PNAS paper. Nathan Cruzado, who will be graduating in May, in case you're looking for a postdoc, is contributed to that paper and a number of other things and has a special expertise in this cross-temporal decoding analysis. And Rifki Afan is actually a student in Ben Scott's lab who's done this really remarkable preliminary work on ALM. Kartik Shankar, this whole Laplace thing was Kartik's idea, and that's a picture of Kartik Shankar, and good job, Kartik. And here's back when we had lab meetings, picture of the lab. I'd also like to thank my faculty collaborators, Mike Halsamo, Howard Eichenbaum, Earl Miller, and Beth Buffalo. I showed you work from them today, and of course, funding agencies. And so thank you very much. All right.
Thank you, Mark. That was fantastic to hear about. And it's really amazing the way that the empirical and the theoretical perspectives in this have come together. It seems one of those fairy tale things in science. It's just really great.
I remember you being really excited about this back then, and you were much more excited than me, actually. I didn't think this was going to work as well as it did.
Yeah, I mean, I love time. I study time and attention. It's been underemphasized, I think, in terms of relative to spatial information, but it's really crucial. We've got a lot of great questions that I think are going to chew up easily the next 30 minutes if we let it. So let me jump right into it. The first one is by Nadine Spatial, and the question is this: is it a good idea to call these time cells? They probably carry way more information than just about time, or is there any sensitivity of these cells with regard to time as opposed to other things they could also be sensitive to?
Yeah, I think, so we have to call them something. I don't think there's such a thing as place cells. I don't think there's such a thing as time cells. I don't think there's such a thing as splitter cells or any of the other types of cells we talk about. The verbal label is a shorthand that we use to be able to talk to each other, so we know what phenomenon we're talking about. I didn't talk about it here, but theoretically, we can construct place cells from the same Laplace transform inverse framework as we use for time cells, right? And we can also imagine a continuity of dependencies in between space and time. Howard and I did a paper in 2015 about this. So yeah, it's a useful verbal convention, but please don't take anything more seriously from that.
But these cells are primarily representing time rather than, for example, featural information about specific events like this light, that square, etc.
I think, well, but they also represent that too, okay. Right? Time cells are conjunctive with position, right? In Chris's original paper, if the animal moves around during the delay, you see cells that fire only when he's in a particular position at a particular time. So in the same way, there's conjunctions for what happened when, there's also conjunctions for what happened where and when. And there's conjunctions of absolutely everything in the hippocampus and the prefrontal cortex. And it's just a verbal convention as to what we choose to focus on. Happily, this Laplace transform framework is pretty general, and we can apply it to many combinations of variables, many variables, and also many combinations of variables. No.
So next question, that was a great one, thank you, is by Matthias Hennig, who asks, would you have suggestions or hypotheses about how long these much longer than the neural time constants may be achieved by the single neurons in these recordings?
Yeah, so Mike Hasselmo and I did a couple of papers about this. First of all, there's a number of different mechanisms. You might imagine that there's recurrent connections with eigenvalues close to 1, right? And you also might imagine that there's intracellular processes based on, like, for instance, ICANN currents that are triggered by acetylcholine. There is such a thing. Angel Alonso did pioneering work on that going back like 2 decades. And Mike Hasselmo is an expert in that. For me, I'm kind of agnostic about the way you build out these equations. I'm really a cognitive psychologist. I think the question of how to build these equations is something that computational and systems neuroscientists should answer more than me. I will say merely that there's a number of different ways it could happen, and I'm partial to the ICANN current story.
Great. Next question is by Jules Brochard, who asks, is the time compressing on slide 5 a kind of efficient encoding?
Oh, yes, absolutely. And that's why we've felt quite strongly about this logarithmic compression for 2 reasons. One is, the analogy with vision and numerosity. But the other question is, why is this true for vision and numerosity? And the answer is that must be something, right? Why is this a common principle throughout the brain? And we've done some work. Kartik and I, and me and Kartik, wrote a couple papers about this a while ago. And the best answer I can give you in a soundbite is basically that because the brain needs to have information about a number of different scales, and it doesn't know a priori what scales are going to be important, right? So if I have A and it predicts some outcome in the future, I don't know if that's going to be 100 milliseconds in the future or like 100 seconds in the future. In order for me to equalize the error that I have for all of those possible scales without placing a strong prior on my expectation for what that scale would be, you end up coming out with a logarithmic compression. And there's a psych review paper from 2018 where we formalized this a little bit. I would also point out that there's a paper up on arXiv by Alexander Shu and Sarah Marzan making a more formal argument that I encourage you to have a look at, rather than me trying to remember everything in the paper.
All right, next up is another great question I'm sure a lot of people are thinking about, is whether these time cells would also work for keeping track of long time intervals, like 1 or 2 hours, or even further. Like, what's the sort of longest range that you think?
Yeah. So first of all, we don't know what the longest range is thus far, because nobody's observed the sequence stopping, right? We've observed time cell sequences going on as long as the delay intervals in different experiments. We have some results, and my student Yue Liu, who's now a postdoc at NYU with XJ Wang, we think we see sequences going on for 10 minutes, but there need not, there's no way on earth you can have activity-dependent firing that lasts for days. Properties of human memory have the same, human memory has the same properties over every scale we can measure, from hundreds of milliseconds up to tens of minutes, up to days and weeks and months. There's no question that there has to be a different type of neurobiological mechanism to go over such long time scales. And we have some thoughts about this, but you can have thoughts too. Our thoughts aren't necessarily that great.
Are you able to, I mean, I guess one of the limitations is that as you get further back in time, the logarithmic compression means that you would basically need more neurons recorded simultaneously to get evidence for
yeah,
that.
that's part of it. But I also, and Mike Halsamo has said this forever, you know, at some point you go to sleep and, you know, the firing changes, right? So I'm relatively confident that you could have a continuity of scales up to maybe half an hour or 40 minutes, something on the order of an experimental session. I think that's reasonable. Things that happen over a longer time scale, you have to appeal to something else. And you have to have something like changing synaptic weights that decay at a variety of rates might be a way to do it. Or have memories recur such that you recycle memories as you go through your day, such that different parts of the brain sample at memories at different timescales or something like that, so that you get this gradient. The equations will work no matter what. So the question amounts to, how do I build a spectrum of timescales? How do I build a bunch of different S's where the time constants go up to whatever upper limit of the scale you're interested in?
Right. I mean, you could imagine though that even like out to 10 minutes as you observe these time cells experimentally, that the firing of those neurons is perhaps being supported by completely different mechanisms, right? Like within a few seconds, it's like active representation, and then you kind of shift over to like a synaptic mechanism, maybe, yeah.
I will say that the Albert, regardless of the mechanism that causes the activity-dependent firing, the cell paper from the Moser lab, they showed exponential decays with time constants out to like an hour, right? So somehow, and it need not be the mechanism we've proposed, but somehow there manages to be activity that extends over many tens of minutes in the lateral entorhinal cortex of the rodent while he's going about his day, that codes for what happened when. And this, in that experiment, it's when did the animal enter a particular enclosure. You can decode that out to very long periods of time.
Do you have a specific proposal about the EC subregions? So the question here is, do you assume that the temporal context cells are, do you assume that the temporal context cells are specific to the LEC and its primate homologs? How do reports of time cells in the MEC fit into this picture?
Yeah, I think what we should is, I think this is a really good question. I think there hasn't been a systematic mapping. In order for me to answer that question, I'd want to know to what extent there's a spectrum of time scales and spectrum of spatial scales throughout these regions. Like, we know there's border cells which behave as if they're Laplace transform of distance to an edge in the environment. We don't know if they have different space constants. That's the first question I'd want to know. We see in preliminary work, looking at data from the Halsamo lab, we see that grid cells are conjunctive with head direction cells, are conjunctive with speed modulation, but that the time constant of the speed modulation has a spectrum. So I think really we need to do careful experimentation with this stuff in mind to try and piece that out. The part I feel strongly about is that there's conjunctions in many variables. How those get assigned to different parts of the entorhinal cortex or the hippocampus or what have you, there's nothing the equations tell us about that.
We got time for maybe a few more questions here. So what is the probability that a neuron representing, for example, t equals 1 also fires at t equals 10. Can a logarithmic distribution fall out of this variability without neurons that are explicitly t equals 10 neurons?
Say this again.
So I guess the question is whether or not this pattern of time cells could arise spontaneously from a probability distribution of the way that neurons fire with respect to time. So imagine, I'm trying to picture this in my head.
Okay. So so the bit about how first of all, they're consistent across trials. So whatever they're doing, it's consistent from one trial to the next. And whatever they're doing, it's sensitive to not only the time at which the trial began. And so you remember the pictures with the rasters that are lined across trials, and they jitter around a little bit, but they're kind of in the same spot. And they also care about the identity of the stimulus that triggered them. So, I mean, in some sense, a spike is an attempt to is carrying some information about what happened when in the past. In some sense, you can think of that as sampling from a probability distribution. But the point is that the probability distribution depends on what happened when. It's possible there's some subtlety about the question that that I didn't gather.
But the fact that they're consistent across trials, I mean, sessions is really important.
Yeah, this isn't just a random phenomenon. We can decode the past, right? We know what happened. It could be useful for memory. Absolutely, we're sure of that.
Yeah. Next question is by Carly Sumbrick. Do you think that it is possible that changes in perception of timing are facilitated by tuning changes in these time cells?
Oh yeah. That fits into the equations really good. So briefly, we can change from time cells to place cells by modulating by the velocity. Like the chain rule from high school calculus. D by dt equals dt dx d by dt. So you can turn stuff, and I might have flipped that. So you can turn stuff by modulating by velocity. You can turn time into space, right? You can get the Laplace transform of position rather than time. And so the way we do that is by having the way that we would do that mathematically. What it means is that you change essentially the rate constant of every neuron in the population together by the same amount by dx dt, and that's understandable as changing the gain on the FI curve of these neurons. So you can imagine something like arousal or some expected time of occurrence affecting the population in the same way. And by changing the gain on these cells, you'd make time go faster or slower, regardless of what the mechanism is that you use to get these slow time constants. If you modulate them all together, you change the rate of time in the network. And so, yeah, we've used that a lot in making place cells and border cells and splitter cells. And we can make a passable model of the diffusion model for evidence accumulation, instantaneous evidence about which alternative you're choosing from. We can build out stuff that looks kind of a lot like populations of evidence accumulator cells in LIP and elsewhere. So yeah, absolutely, positively, it can be done. The big question, it comes back to this question earlier about MEC. If I'm at this point kind of convinced that these ideas make sense of a ton of stuff, right? But there's nothing in the equations that tell you how to fit the parameters, how to choose what is the gain for a different population, or whether this is modulated by velocity or this is modulated by the rate of information accumulation or expectation, or whether these cells are responding to pictures of cats and dogs and these other cells are responding to odors, and how those are distributed or flexibly changing across trials. So the theory doesn't tell us anything about that. And I think we should, I think we as a group should apply, we should take these sort of ideas and look through the lens of this this nascent theoretical framework of the data we have in hand, and think about new experiments to map out the brain.
Yeah. Well, thank you, Marc. That was fantastic. I love hearing.
Thank you for having me. Thanks for the great questions.
Ideas for things that bring everything together and might actually work, which is awesome. I think this is one of those. Yeah.
Good
All right.
Great to see you.
All right.
Hope it passes in real life.
Yep. Bye, everybody. Thanks for coming.